rapid oral presentations. Our first presenter today is Katie Rogers. Katie is a fourth-year PhD student at the University of Florida under the guidance of Dr. Jose Chaparro in the Fruit Tree Breeding and Genetics program. Her research is focused on characterizing flowering in citrus with the goal of developing biotechnology applications to shorten the generation time in citrus and accelerate breeding efforts. Katie grew up on a citrus and cattle farm and in Wauchula, Florida, and received her bachelor's degree in horticultural sciences, plant molecular and cellular biology at the University of Florida. So please join me in welcoming Katie Rogers. Presenter today is Mahendra Bhandari. Mahendra is a second-year PhD student working under Dr. Amir Ibrahim and Dr. Qingwu Xu in wheat breeding and crop stress program. He received his bachelor's degree in agriculture science from Tribhuvan University in Nepal. He completed his master's in plant, soils, and environmental science at the West Texas A&M University. His current research is in the use of high-throughput phenotyping system in wheat breeding with a focus on disease, yield, and water stress phenotyping. So please join me in welcoming Mahendra.
Is it okay?
Yep, I got it.
Thank you for the nice introduction, Nolan. And first of all, I'd like to thank the Symposium organizing committee for providing me this opportunity to talk about my research. And today I'm going to talk about using aerial imagery to assess the wheat foliar disease severity. And we heard a lot about phenomics and using remote sensing in agriculture. And I will be more specific on aerial imagery and disease severity. And particularly, I'll be talking about the wheat leaf rust. Leaf rust is caused by a fungus Vaccinia and is a major disease of wheat. And when the plant is infected, small brown pustules are developed on the leaf surface. This reduces the chlorophyll content of the leaf, which provides an opportunity to use remote sensing for differentiating the diseased vegetation and the healthy vegetation. You can see in the picture, in the graph over there, I'm sorry. Over here, there is a contrast in the visible and near infrared region of the electromagnetic spectrum when the sensors are exposed to the diseased leaves and the healthy canopy. So we are catching up that contrast to see if we can identify the disease and see the prevalence of the disease. And the major objective of this research is to see if we can use this imagery technology to identify the disease and evaluate the disease on different genotypes. For this, we flew DJI Phantom 4 Pro, which is relatively cheaper drone, and was mounted with RGB camera to take the RGB images. And the research was conducted in Castroville, Texas. For field data collection, we collected the data on the basis of visual ratings for infection type and disease severity. Infection type comes with the susceptibility ratings, which might be susceptible, moderately susceptible, moderately resistant, and resistant. And the severity notes were taken on a scale of 0 to 100. The infection type was later converted into the numerical value and multiplied with the severity notes to calculate the coefficient of infection. And on the image processing side, what we did is process the image to develop excess green index and band ratio. Excess green index, what it does is it highlights the green portion of the canopy in the plot. And the band ratio it highlights the diseased portion of the canopy in the plot. And we also tried several other image classification approaches. The image over here, it is the orthomosaic image of the entire field, which is almost 20 acres of the area. And for this study, we are focusing on mainly on this corner of the field, which consists of 40 different genotypes and 3 replication. And the next picture over here, it consists of the plot boundary layers, which is used to extract the data from individual plots. With that approach, I'd like to move to the results. The results shows that both of those indices, excess green index and bend ratio and the maximum likelihood classification approach, was able to identify the diseased portion over here and the healthy vegetation. The first picture over here is the normal RGB image, and it consists of, here are the little plots that are surrounded by the black boundary. Those are the plots that consist of susceptible varieties. And the plots that are surrounded by the yellow boundaries, those are the plots which are resistant varieties. So what these indices did is they were able to isolate those two different plots based on the severity of the disease in the field. And after extracting the data from the field, we could see that the excess green index and the bend ratio both were highly correlated with the coefficient of infection. So to summarize, we can say that this simple technology of aerial imagery can be helpful tool for high throughput phenotyping, particularly for disease phenotyping. And what important point we need to focus is that how automated is the process of image processing and data extraction. And the take-home message from this is a simple camera and a simple drone can provide a lot of information regarding the plant behavior so that the physiologists or plant breeders can use for better study of the plants and make rational decision during the selection program. And I'd like to acknowledge my committee chairs and everyone who was involved in this project. Thank you. And I'd love to have the questions if you have any.
We do have several minutes, so if you raise your hand, we'll get this one.
So how did you had 3 approaches up there, the maximum likelihood approach, how'd that do?
It is like what we do is, I used NB for using that maximum likelihood approach. What it does is it classifies the image based on the data that we supply to the image, like supervised classification. It was better. Actually, I tried several other approaches of image classification as well, and it was relatively better than other ones.
I have a question. So when I talked to the wheat breeders, they said that sometimes with the resistance, the resistant variety does a little more yellowing, though the spots are much lower. So the severity, it tries to maintain, it looks sick, but it tries to fight out the disease much better when it comes to the resistant variety versus susceptible. So how did you see that in your...
Yeah, actually that is, I think you were talking about this earlier as well. We know something is going on in the field based on the image or based on the indices, but we are not quite sure actually. It might be because of physiological changes or maybe senescence or something like that. What we found is like those ratings were highly correlated with the visual ratings. So we still think that there is a chance of improvement, maybe being more precise for pixel selection. Let us see, say if we select the particular pixel of that brown spot, then use that particular spot as a ground truth to classify the image. maybe that would be the better approach. And we'll be using that approach in the coming season too. It is just the RGB image view, and we are increasing the number of sensors as well, multispectral and thermal cameras. So maybe we can just stack them together and find out something.
Yeah, your correlations are very good actually. So, yeah, I don't see a need for multispectral if you can
Yeah,
get the similar data.
yeah, that is what I wanted to focus is like, this technology is really cheap, like maybe around 2,500 or so, the Phantom 4 Pro and the camera. So multispectral will cost like $5,000 or $6,000. So maybe we can stick up in this cheaper technology and do the same job what other sensors can do.
Thank you.
All right, I think that is all the time we have. So thank you, Mahindra.
Thank you.
All right, our next presenter today is Nicholas Ace Pugh. Ace is a doctoral candidate in Dr. Bill Rooney's sorghum breeding lab. His research focuses on the utilization of remote sensing techniques for high throughput phenomics and in the implementation of these technologies into a breeding program. Please join me in welcoming Ace Pugh.
Thank you, Nolan. So as Nolan said, I'm Ace, and today I'll be presenting a project that actually is the product of a pretty large interdisciplinary effort across the university. And that project is temporal estimates of crop growth in sorghum and maize breeding enabled by unmanned aerial systems. So as I said, we have a large collaborative effort in this university to try to use UAS for various agricultural applications. And this is one of the fruits of that labor. Now, when we speak about evaluating UAS for their applications towards agriculture, there's a difference in opinion, or maybe just a difference in values between plant breeders and engineers and computer scientists. Neither viewpoint is wrong, but there's a difference in applicability depending on how you look at it. So engineers are usually going to be concerned with correlative analysis and having the highest R-square value that they can get. And that is important, but as plant breeders, we're actually usually more concerned with being able to tease out genotypic variation and implement these technologies within a breeding program. So these are just a few of the results that are presented in a recent paper on this subject. And I want to draw attention to the left side of the screen with the variation explained figure. Now, we did this study using structure from motion. That was how the UAV height estimates were derived. And we also used ground truth measurements for comparison, and those were recorded using the traditional method of a height stick. So you can see here that the variation explained between two different sorghum tests and two different maize plantings is pretty high via the UAV. Actually very similar to ground truth methods and even superior in some cases, especially early in the growth season. It's also important to draw attention to the fact that it does kind of depend whether you're measuring sorghum or maize or any other crop, what type of measurements, what portion of the point cloud that you use. So here we showed a couple of different percentiles of the point cloud data that was generated from structure from motion. So in this case, we have the max percentile and the 95th percentile. And as you can see, there is a difference between the two. But overall, the UAS are actually very good at explaining genotypic variation, and that's really important for breeders. Now, we also wanted to take this a step further and look at maybe a way that we could actually implement this technology and maybe derive new phenotypic information from it. And I want to draw attention to the right side of the screen with these high-resolution multi-temporal growth curves. Now, the sorghum growth curves, A and B, were generated just using a single UAV in Corpus Christi, Texas, using a Phantom 3, I believe. And C, though, the maize growth curve was actually generated using that same Phantom and two other UAVs, an EB and a Solo. So what's kind of interesting here is that we can actually derive some information that you maybe wouldn't have access to if you only took a final plant height. You can see different crossovers between genotypes that maybe occurs early in growth, which could be an estimate of plant vigor or response to stress. So right now we're just showing this kind of more in a qualitative sense, but there's probably going to be some applications soon where you try to utilize this information quantitatively as well. So future objectives for this work, I would like to kind of assess UAS for their ability to phenotype other traits of interest in sorghum. So a couple examples would be biomass yield or disease scores. And also even generate maybe curves for those traits as well, because different valuable information could be gleaned from that. And finally, I would like to also integrate these UAS height measurements into standard plant breeding studies and start to look for within plot variation. So 5 minutes isn't a lot of time, but I think I gave you a good overview of what you'll see if you come visit my poster. This project was also recently published in the Plant Phenome Journal. This is a list of most of the individuals and organizations that made this research possible. It's not an exhaustive list, though, because a lot of people worked on this project. And I would also like to thank the Symposium Organizing Committee for putting this on today and for giving me the opportunity to speak here today. And with that, I'll take a few questions.
Thank you. We have about 5 minutes for questions. Yes, Dr. Zong. Here, I can. Oh, we got one coming to you.
I have a little general question. Question that there's really, so far there are few type kind of technology for breeding, like a marker system breeding, genomic selection, right? There's certainly current traditional breeding, phenotypic selection, and now that the high throughput phenotyping. If you make a comparison to the technology, what's the advantage of high throughput phenotyping technology?
So the
over the current other technology, genomic selection, marker selection, and traditional phenotypic selection.
So what are the advantages?
Yes.
So I think the best comparison here is between high throughput phenotyping and regular traditional phenotyping, because in that case, high throughput generally is going to be more useful if you're analyzing a large number of plots. If you want the most, I guess, accurate measurement possible, you probably would still want to use a height stick on a ground level just because you know that you're getting the absolute best. But for most breeding studies, I could see it's being preferable to that method in general. As far as comparing it to things like genomics, I don't really necessarily think that they can be compared in that fashion, because really I think you would utilize this in tandem with those technologies. So I think that they just complement each other, really.
We have one at the back. Dr. Murray.
So since you didn't mention it, the growth curves near the end decrease. And I think many people have discussed with you why they think that is. Why do you think that is at this point? And you know, why is that important?
So the answer to that, I guess the conservative answer would be that I don't really know. So I don't want to make declarations that aren't necessarily backed up. But so I think if I was to hypothesize, I would say that's probably due to a large number of different factors. So we know that plants senesce later in the growth cycle, and the heat can also cause droopage of plants, and there's a lot of other things, lodging. So there's a lot of answers for why you could see this decrease. Now, another possibility that would need to be investigated is that maybe plants actually have stalk shrinkage later in their growth cycle. But that would be something we would need to investigate before we make such a claim.
No, it's actually from me. So, based on this work, do you think we would be able to know which growth stage or what point in the life cycle is best for phenotyping, for vigor, or like when it's indicative of yield?
I think that's maybe an application that will be developed, because that's kind of, the growth curves kind of gets at that, and different genotypes might actually have different points where that response happens. So when you take measurements many times over the course of a season, for example, these growth curves that you see here were generated from somewhere between 15 and 20 different flights, depending on the crop. When you have that kind of information, you can really see where each genotype actually had this response. So that's kind of one of the values of having this sort of a qualitative representation of plant growth.
That's all the time we have, so let's please thank Ace.
Thank you.
Just a reminder that if you would like to hear more about any of our presenters' projects, they will be available next to their posters in the upcoming poster session. So next up, our next presenter is Suraj Sapkota. Suraj is a PhD student at the University of Georgia Institute of Plant Breeding, Genetics, and Genomics. He's working on wheat diseases, leaf stripe, and rusts for his PhD research. His research emphasizes the identification and characterization of the genetic basis of of resistance to leaf and stripe rust in soft red winter wheat cultivars. He received a Master's of Science degree from the North Dakota State University in 2015. His master's thesis was titled The Identification and Genomic Mapping of Resistance to Bacterial Leaf Streak Disease in Wheat. He received a Bachelor of Science in Agriculture from Tribhuvan University, Nepal in 2010. And with that, let's please welcome Suraj. Thank you.
Yeah. All right, thank you so much. At first, I would like to thank the organizing committee for providing this opportunity to come here and present my PhD research work. So today I'm going to talk about one of the major QTL that identified for the leaf rust disease in wheat. So wheat is a major crop worldwide, and large proportion of world population depends on wheat. So about 20% of calories that we need for human population that comes from wheat. US is a major producer of wheat, and it contributes about 10% of world production. There are about 42 states in the US they produce wheat, with Kansas and North Dakota as the leading states. Leaf rust is a major disease. There are three different kinds of rust: leaf rust, stripe rust, and steam rust. So leaf rust is the major one with wide distribution and most common disease. So it is also a major disease in the southeast, a major disease of wheat in the southeast, and up to 40% yield reduction is possible because of leaf rust. So one of the major problem with leaf rust is that this pathogen continuously evolves new race. So on an average, from the US, over 50 races are reported every year. So that demand us to continuously search for a novel source of resistance and novel gene. That is the reason my objective of my research is to identify QTL that we can use in the breeding program to develop leaf rust resistant wheat variety. So initially what I did was I evaluated the parents with these three most common races of leaf rust, and I found that the AGS 2000 is resistant to all the three races. However, this Pioneer 26R61 parent is susceptible to one race and resistant to two other races. So in this photo, you can see the reaction of the two parents with the leaf rust ratio 1. So this is highly resistant reaction, and this one is susceptible reaction. So I choose this race and screen my real population. So I have 175 real population developed from the cross of these two parents. And this graph shows the distribution of real population to each disease category. So we see either resistant or susceptible category. There is no any intermediate reaction. So that indicates that there is a single major gene conferring resistance to leaf rust. So for the mapping purpose, I used a genetic map constructed by Howe et al. So the map includes about 2,800 markers. So the markers were SSR and SNP and dot marker. It is a kind of integrated map. So that map covered the whole genome of wheat. And from the QTL mapping work, I identified a major QTL on 2B chromosome. So this is a highly significant QTL with log value of almost 14, and the R-square value is almost 27, 28%. So I identified the QTL, but the next question is this a novel QTL? Because there are more than 70 genes identified for leaf rust. And the question is, yes, this is a new QTL. So here is the evidence. So there are two other genes reported from the 2B chromosome. So LR50 and LR58. So if we see the origin of these genes, so LR50 was originated from the wild tetraploid wheat, and LR58 was originated from barbet goat grass. It is highly unlikely that these genes are present in the common wheat. And another evidence is that if we see the physical location of my QTL with other two genes, we can see, so the LR58, so LR50 is located in this deletion bin of the physical map, and LR58 is located in this deletion map, and my QTL is located in this deletion bin. So this also indicates that my QTL is different than the previous one. So the conclusion of my research is, yes, I identified a QTL which is novel, and we can use that QTL in wheat breeding program to develop like leaf rust resistant wheat cultivar. With that, I would like to thank my major professor, Dr. Muhammad Marzoon, and I also would like to thank Jerry Johnson and Howe. So Howe is the person who developed this mapping population and did most of the genotypic work. I would like to thank Dan, Steve, and Ben for helping me in my research. With that, I would like to take questions if you have. Thank you so much.
Thank you, Suraj. We have 4 minutes, I believe, for questions. That wants to raise their hand. Any other questions? Yes, Dr. Klein. We'll be right there.
So this was for one race, but are you building like durable resistance by pyramiding, genes for? You mentioned using it in a marker-assisted selection breeding program, but would you need to have QTL for the other races of resistance to be able to do that as well?
Yeah, this QTL is for the race 1. So this race 1 is the most common race that we have in Georgia. I'm also screening my parent with other race from other states, and I haven't found any difference between the parent. So I'll continue doing that with almost 10 or 15 races of leaf rust. And it is highly possible that we can use this QTL and integrate with other QTL to develop a broad spectrum resistant wheat variety. Thank you.
We have a question from Dr. Murray.
You're planning on using this in plant breeding or marker-assisted selection. Is there any interest in the lab that you're in to try and identify the genetic basis of this gene, or is that just extra work? And what's your goal?
I think that's what I can do for my time right now. But the person who is going to follow up this work, he may be working on fine mapping and narrowing down the QTL region.
Okay. Any further questions? All right. Well, I guess with that, let's thank Suraj.
Thank you so much.
Our next presenter today is Anna Castro. Anna is a fifth-year molecular and environmental plant sciences, or MEPS, PhD student in the Mullet Lab. Her research focuses on the genetics and physiology of sorghum stem development. She has previously served as the president of the MEPS Students Association and the chair of the annual MEPS Symposium organizing committee, which is a lot of work. So please, join me in thanking Anna Castro. Thank you so much for that introduction, Nolan. My research focuses on sorghum stem development and composition, and you may be asking yourself, why should you care about sorghum into bioethanol production in several different ways, as you can see here. The grains, the juice from the stalk, and the stem and leaf material itself can all feed into bioethanol production. The ideal sorghum biofuel crop might be optimized for two or more of these traits in order to feed into bioethanol production in multiple different ways. The stem here is the main organ for accumulation of biomass and stem sugars in sorghum, but the problem is that high biomass accumulation and high stem sugar concentration may not necessarily be present in the same variety. A problem is that some varieties of sorghum selectively destroy their pith parenchyma cells, effectively hollowing out their stem, and you can see that from this progression. You can see that progression in this photo here, or in these CT scans. So the sorghum germplasm shows a lot of variation for this trait that we call arrhythma formation, and so we call this arrhythma formation. And even though this trait has been selected against in a lot of sweet and grain sorghums, we don't actually, until this point, have known much of the genetic mechanism or regulation underlying this trait. So what I've done in my research is I used the genetic resources available in sorghum to map this trait, variation for arrhythma formation, in 3 biparental populations to a locus on chromosome 6. I was then able to find map that locus to a single gene encoding a NAC transcription factor that has a single nucleotide polymorphism causing a stop codon and a truncated protein. Further expression studies that I have done show that this NAC transcription factor this is the genotype that does form arrhythma, and this is the genotype that does not form arrhythma. You can see that expression remains mostly the same as you would expect since this is non-functional at the protein level. However, these genes, several genes involved in programmed cell death, you can see are highly upregulated in the stems of genotypes that do form arrhythma compared to those that don't. A lot more work needs to be done to understand the regulation of this gene and how other internal and external influences will affect its expression and downstream arrhythma formation. But by identifying this gene, we've taken, I think, a significant step forward in designing the ideal biofuel feedstock crop. So with that, I'd like to thank my committee members and my advisor, Dr. Mullet, and all my former and current lab members, as well as the Rooney Lab, and the numerous people that have helped me in the greenhouse. And also, I'd like to thank my funding resources, the GLBRC, and the Terry Atkinson Chair. I'd love to take your questions. Oh, and my poster number is number 17, so I hope I can go into a little bit more detail of my research there. Questions? All right, we have 7 minutes for questions. However. Yes. Here. Awesome. Maybe I misheard you. Did you say you did the PTO mapping and then you went all the way down to the single gene? I did. How'd you do this? So what I had was I had several populations that were real populations that were already available, and as soon as I started my PhD, I mapped that trait, and actually several other people in the lab had also mapped the trait. But the population I went back and mapped it in was quite a bit larger than the other 2 that it had been previously. This PTO had previously been mapped in, so that just gave us a little bit more confidence. And basically, I got a little bit lucky in being able to, in the breakpoints that were available within the markers, so that I was able to narrow it down to quite a manageable region, and only had to do 1 or 2 rounds of fine mapping before I was able to identify a small enough region that had a manageable number of candidate genes. We also had whole genome sequences of these parents, so I was able to compare those candidate genes, the sequences of those candidate genes directly. Very good. Roots can form arrhythma too. Do you think this gene is involved in arrhythma formation? It's possible. We have an expression atlas within sorghum. The problem is the genotype that this expression atlas is in is a genotype that doesn't form arrhythma, which might be okay. But we think that it's possible that both stem arrhythma and root arrhythma might be, this gene might be involved in both mechanisms, and that's definitely something we'll be working on checking in my next several experiments. Any questions? Does this trait make it much more prone to lodging, or? I'm not sure. I think there have been some studies on sorghum stem lodging. I don't recall if they found any significant interaction between lodging and arrhythma formation. Some people think that it's been selected against in grain sorghum by, not directly, but indirectly by selecting against the lodging. But I've read some other studies that indicate the hollower stems may be more stable. So we're not sure yet, but that'd be something interesting to look into. You have a working document. So the classic way to prove the function of the gene, the last step would be to do the transgenic, or not or something like that. Do you guys plan to do that? Our lab is working on some sorghum transgenics, as I'm sure people are aware. It's transforming sorghum a little difficult. But one experiment I'd like to try to do is, since this is since the non-functional version of this gene causes the solid stems, I'd like to do basically a complementation of that gene to see if I can get those stems to also form arrhythma. Yeah. Any other questions? Any further questions? All right. Well, with that, let's thank Anna. Thank you so much. All right. So thank you to all of our rapid oral speakers today. We have now dedicated an hour today to solely poster viewing. The students that brought their posters today are studying plant breeding and other related sciences. We encourage people from across the university to participate in today's events. So we have 20, that's the newest count, 20 posters for viewing today. They are in the room behind me, behind the projection screens. This morning, the posters were judged by a panel of plant science experts. Each student presented a quick summary of their research to a subset of the judges this morning. Judges were asked to select their top 3 poster presentations, and scholarships will be awarded in the amount of $200, $150, and $100 to first, second, and third place, respectively. We ask that all students remain at their poster for the duration of the poster session, and we encourage the audience members to interact with the students and get them to have a chance to explain and present their research. So I would just like to reiterate, this poster competition is only possible through the donations of our sponsors. So thank you so much. Along those lines, we also have snacks, coffee available at the back of the room. Lunch will be served at 12:30, and this is very important. So it's this hallway back here. There'll be tables back there, and you can walk through. I think we're going to start on that side. So starting at 12:30 today, we'll have a catered lunch by Jason Stone, is that right? Yeah, Jason Stone. So for those of you on the webinar, the next presentation will be from Dr. Kirti Rathore, and will begin at 1:40 p.m. Central Time. We encourage you to share your experiences on our event, on our several social media platforms, and request that you tag us on these platforms. Once again, we would like to thank our sponsors because their support made lunch, snacks, poster awards, travel scholarships,